I'd like to introduce the last speaker of the symposium, Dr. Dan Martin, who's the chair of Cleveland Clinic Coli Institute. And the title of his talk is Unanswered Clinical Questions in AMD, Clinical Trials We Did Not Do in the DRCR Retina Network, and Why. Dan. You're on mute, Dan.
Thank you. Anyway, thank you very much to the Cleveland Eye Bank Foundation for putting together this symposium, and also for your ongoing support of all three of the institutions who are represented here today. I'm going to, I have the same problem Stephanie was having. That's interesting.
Use the monitor rather than keyboard.
We're good. No, we're good. We're good. We're good. I got it. So I have no financial interest to disclose. So last year at this meeting, I presented sort of the top 5 greatest hits from the DRCR Retina Network, which is a large multi-center randomized clinical trial group sponsored by the National Eye Institute. I serve as the chair of that Retina Network. It's the largest clinical trials group in ophthalmology, along with Jennifer Sun at the Joslin Diabetes Center at Harvard. We have over 500 retina specialists who's part of this, and 1,800 other personnel who support the many clinical trials that we have performed over the last 20 years. We've completed 35 studies. We have 5 that we're reporting out. They're either ongoing or reporting out now. We're going to launch 4 more. The initial 15 years of this network was all focused on clinical trials and new treatments for diabetic retinopathy. But the last several years, we've expanded our scope such that we now study all retinal diseases, and there's been a strong focus on macular degeneration. Each one of these studies begins as a protocol idea, oftentimes presented to the network as a potential trial by one of our investigators. It goes through an exhaustive process. It's reviewed by our operations group if we think, and then it's sort of filled out at that point. At that point, it's presented to our investigative group. And then it goes to the protocol development committee. It's got to pass each one of these sort of stages. Then it goes to the executive committee to approval, goes to approval of the data safety monitoring committee. Then there's an external protocol review committee at the NIH, and ultimately you wind up with a final DRCR network protocol. This process takes about a year. It's exhaustive. It goes through many, many levels of review. Quite frankly, it's probably more rigorous than a lot of study section reviews, to be honest. But the whole thing starts out with a protocol idea. Over the last 12, 13 years, we've evaluated over 242 of these protocol ideas. Many are common clinical questions encountered in day-to-day retina practice. Each is carefully reviewed, and some are extensively developed. Very few of these ideas ever make it to a full trial, only about 1 in 20. And it represents a very large body of academic work that no one ever sees. So I got the idea of putting this together because we kept getting a lot of the same protocol ideas. A lot of people would think of the same ideas and wonder, can't we answer this clinical question in some sort of study? And so I thought it might be helpful to give you an idea of the process that we go through and the good ideas that were presented, but why we didn't do the studies. And it'll give you an idea of just how exhaustive this process is, and again, really how it's an amazing academic process. So the first question, and I just chose 3 from age-related macular degeneration. So this was a question that came up probably 3.5 years ago. A lot of pharmaceutical companies were very interested in beginning to inject the fellow eyes of patients who had neovascular AMD who were at risk for progression. So in this panel, you see the right eye has neovascular AMD, the left eye has high-risk drusen. And the question is, should we be treating these eyes prophylactically, we'll say with intravitreal Avastin or aflibercept or ranibizumab? And so we spent a lot of time with this one. This one we probably spent more than a year on this one. It actually got assigned a protocol letter. And we thought we were going to do this study. And we had a lot of people who were supporting us in doing it. It was a very appealing idea. Again, you have bad disease in one eye, the fellow eye is at risk, an injection every 3 months, possibly to prevent it. And so that's what the trial was. About 1,000 eyes randomly assigned to an anti-VEGF drug every 3 months, an anti-VEGF drug every 6 months, or sham treatment. The primary outcome was development of choroidal nevascularization in the study eyes confirmed on FA and OCT. We got towards the tail end of this thing, and we kept asking, what exactly are we preventing? And we wound up looking back at the CAT data. And we've published 65 papers out of CAT. You would think we had looked at this question before, but we were kind of shocked that we hadn't. And one of the things that was very interesting was about 3/4 of the eyes came into CAT with unilateral neovascular AMD. Over the course of the study, about 10% per year developed neovascular AMD in the fellow eye. And what's interesting is that the baseline visual acuity that they presented with was better than when they had presented in the study eye, 2032 versus 2062. When we looked at 1 year, how well did those eyes do? They actually did pretty well at 2032. And then here's the distribution of the fellow eyes in CAT. And at the end of 1 year, 85% of these eyes were 20/40 or better. So a couple of our biostatisticians within the DRCR network, notably Maureen McGuire, put together this predictive model based on the CAT data and several other sources. And when we ran the model, what we came up with was that if we did no preventative treatment, the proportion of eyes that should wind up better than 20/40 is actually 95%. And that if you were 25% effective or 50% or 70%, you really didn't change this that much. But the cost of doing this was extraordinary, that the number needed to treat was over 20 to benefit one patient. And the cost of doing that for the entire population would be $1.2 million to prevent one eye from winding up less than 20/40. At the same time, there were 2 small pilot studies that were ongoing, one looking at Eylea as prevention and the other one as looking at Lucentis ranibizumab for prevention. And what's really interesting, neither one of these trials actually worked. Showed that with these injections given every 3 months in both settings, that there was no difference in the rate of CNV conversion at 1 and at 2 years. So again, gives you an idea just how sort of detailed of analysis we will do before we launch into a trial that costs maybe tens of millions of dollars. The second question is, should ARED supplements be continued when the second eye develops neovascular AMD? So again, in the left panel, you have neovascular AMD that's been longstanding. In the right panel on the left eye, you have a new choroidal neovascular membrane. So what really happens in clinical practice is we get very caught up in treating the left eye with anti-VEGF therapy, and we sort of forget. And about 3 or 4 months into it, the patient will ask you, "Gee, should I continue these these ARED supplements?" And so it was a great question. It was actually submitted as a protocol idea by several of our investigators. So to assess whether or not we should do a trial looking at this, first you have to go back and look at what were the primary effects of ARED supplements. Well, they were to reduce the risk of progression to neovascular AMD, which it did about 20%. We still don't understand the mechanism as to how zinc and antioxidants would do that, but that's what was shown in AREDS 1, and also reduce the risk of vision loss by about 25%. So the problem is, in these eyes, both AREDS outcomes have already occurred. So what would be the primary outcome if we were to do a clinical trial? Traditionally, we like to use mean change in visual acuity, but the problem here is that the effect in AREDS was not large, and we're about to start anti-VEGF therapy, which is going to significantly mask any sort of visual acuity effect from the vitamins. And so if there is an effect, it would take a giant trial to show a difference. We could look at an anatomical outcome growth of CNV, but that's challenging due to the variable growth rates among eyes with choroidal neovascularization. There's a low coefficient of determination, R-square, with correlation between size of the lesion and visual acuity. So we elected not to do that study. This was an idea that was submitted also multiple times independently. And that's the question is, what's the optimal management of a large subretinal hemorrhage in eyes with neovascular AMD? So we encountered this problem from time to time. It occurs in about 1% of all cases or less. And we took a pretty exhaustive look at this trial, potential trial 2, and concluded it would be a real challenge to enroll because of the frequency of the disease is not common. And the other is that there's a wide variability in presentation. So you'd have a challenge in trying to define this. I mean, all these different cases, and I could show you lots more, trying to determine exactly how much hemorrhage we would require the geometric center of the fovea, how much of it had to be subretinal versus sub-RPE, how thick did it have to be. A lot of things that are really difficult to quantitate objectively. And when we began to kind of work through those eligibility criteria, it was going to be a real challenge. The other problem that we had is that we already know that some eyes do very well with anti-VEGF injections alone. So here is, again, data from CAT, an eye that presented with a large central hemorrhage, and simply treating this eye with monthly injections, the eye winds up 20/25, 1 year later. The anatomy doesn't look great, but the eye still winds up 20/25. Here's another one that started 2100, finished 20/25 just with injections, no surgery. Sorry, the comparison was going to be anti-VEGF injections versus subretinal surgery. We used to do surgery on these eyes all the time. I did a ton of the surgery. And nowadays, honestly, I haven't done this in a long time, because even these large hemorrhages will respond very well to anti-VEGF injections. So, but there are still investigators who want to do this. And so as we began to talk to our investigator group, we realized we have a huge problem with investigator equipoise. And then there was the issue of the public health impact would be low when balanced against the limited resources and the $10 to $15 million it would cost to do the trial. So the reasons for not conducting a clinical trial, these were just 3 examples. One was the estimates of clinical effect are low, and the number needed to treat would be unacceptable and cost excessively high, as I showed in the first example. The second example, with the area of supplements, should we continue them, the challenge was defining the primary outcome. In the third example, we had a disease that had a low prevalence with variable presentation. You had challenges related to enrollment, defining the entry criteria, and achieving investigator equipoise. And then finally, when we look at all these trials, we're constantly trying to balance the public health visual benefit against the cost of the study. Thank you very much for organizing this symposium. I'm happy to take any questions.
Thank you, Dan. We have one question on the chat that I'll ask, but while I'm asking that question, maybe I'll ask all the panelists to turn on your video, just in case there are other questions. So the question, Dan, is from Mark Lackey. What causes subretinal hemorrhage AMD?
Oh my, if I knew the answer to that. It's a fantastic question. So you would think that we would know the answer to that. You would think, and just to tell you how poorly we understand that, you would think that in patients who are anticoagulated, that there would be a higher risk of subretinal hemorrhage. Very intuitive that would be the case. Turns out there's not. We've shown that, it's been shown in lots and lots of studies, that the presence, people who are anticoagulated for, say, AFib or whatever reason, that they do not have a higher rate of subretinal hemorrhage. So I have no idea. That's the honest answer.
Okay. I can start off with a question. I'm not seeing any on the chat. Dr. Peachy, in your